This is an A-level biology presentation on the uses of monoclonal antibodies. In this presentation, we're going to look at what a monoclonal antibody actually is. We're going to look at examples of how they're used in terms of medical diagnosis and targeting of medication to specific cells. We're also going to look at the ELISA test, and we'll have a mention about the use in terms of ethics of monoclonal antibodies. First thing you need to be able to do is identify the different regions of an antibody. So pause the presentation, work out as best you can 1, 2, 3, 4, 5, 6, and 7. And then when you're ready, you can restart the presentation and check your answers. If you don't know numbers 1 to 7, what I suggest you do is look at the answer straight away, spend some time memorizing it, and then go back over at another point until you can identify them all from memory. So pause the video now and then restart when you're ready for the answers. So let's go through the answers. Number 1 is an antigen. 2 is an antigen binding site. Note that number 1, obviously being an antigen, is normally attached to the surface of a membrane or surface of a pathogen or surface of a cell. We have 3 is what's referred to as the variable region. 4 is known as the light chain, again because it's smaller in size. We also have what's called a constant region. The constant region is common to all antibodies. It is that section number 3, which is the variable region, which has the antigen binding site that actually is different depending on which antibody you have produced. We have the heavy chain, which refers to obviously the longer section on the antibody. And then finally, we have the receptor binding site. And that's where we have binding of anything to an antibody. So if we want to attach some sort of marker, or if we want to attach some kind of enzyme, that's the section that we would attach it to. Antibodies are made up of a number of polypeptides. They have 2 light chain and 2 heavy chain polypeptides. They have complementary regions, and at one end we have what we refer to as the antigen binding site, and the antigen binding site only binds to a particular antigen because those shapes are complementary. What we then have at the opposite end is the receptor binding site, where we can add on a range of different receptors, and we'll look at that in due course. The term monoclonal antibody is in reference to the fact that each antibody is an identical copy as long as it has come from the same B cell and is a result of the same pathogen. So in terms of antibodies, they are all identical, hence the term monoclonal. Now there's 2 ways in which antibodies work in terms of the immune system. And the 2 pictures represent those 2 ways. Pause the presentation, have a think about how the immune system works, and think of what the 2 functions of antibodies may be in the immune system. Pause the video and restart when you're ready to look at the answers. So the 2 processes that involve antibodies are, one is agglutination, also known as clumping, and this is to make the phagocyte's job easier in terms of the immune system. And the other thing they can do is they can act as basically markers, and they basically encourage phagocytes to attach and target the particular pathogen. Now we have 3 uses of antibodies, and all 3 uses are linked into this concept of the antigen and the antibody are complementary shapes. So that antibody will only attach to specific antigens. The first example we've got is the targeting of drugs to cells. So we have what is called direct monoclonal antibody therapy. And a direct monoclonal antibody therapy, what we use is antibodies to target particular antigens that are found on cancer cells. And what they do is they stop uncontrolled replication. So they're directly blocking any chemical signal passing to the cancer cell. Now we also have this thing called indirect monoclonal antibody therapy, and I want you to pause the presentation, have a think about what that may be, looking at the diagram on the right hand side, And when you're ready, you can restart the presentation. So pause now. So indirect monoclonal antibody therapy is essentially where we attach some sort of radioactive or cytotoxic drug onto the particular antibody. It means that when that antibody binds with the antigen on the cancer cells, it actually delivers that radioactive or cytotoxic drug, causing destruction of the cancer cell. Now, there are pros to either side of the direct monoclonal or indirect monoclonal. You need to be aware of each one, and you need to be why each one would be used in terms of its positive impact. Second application is in medical diagnosis. What we need in medical diagnosis is a rapid and easy method that minimizes the risk of false positives and false negatives. You want to make sure that if you say someone's got something, you're 100% right, and vice versa, if you say somebody hasn't got something, you're 100% right. A lot of the time the mistakes in diagnosis come down to not the actual test, but the person reading the test and interpretation of the data. So you try and take that away, the human element away, to reduce the amount of error. In this particular example, there is a protein found in a patient's blood. And that particular protein could be a pathogen, it could be one of another of other substances that you're testing for. And what you've got is, in a lab, you've created 3 antibodies, and we've attached 3 fluorescent markers, A, B, and C. And what you do is you basically apply A, B, and C to the sample of patient's blood, and what will happen is that antibody C is complementary to the antigens on the patient's blood for that particular protein, and so will bind. So when you wash the sample and clean the sample, A and B will be washed away, and you'll be left with C, which means that the sample will show up in terms of the bluey teal color. What you can also do is you can also talk about it in terms of intensity, because the more blue teal color you have, the more of the particular protein you find in the blood. One of the other common uses of monoclonal antibodies is the use in a pregnancy test. To start with, we're going to look at the structure of a pregnancy test, and it's basically made up of 3 separate regions. We've got a reaction site, a test site, and a control site. On the reaction site, you have antibodies that are attached with an enzyme on the end, but the antibody is not attached to the test strip. It is free to move along the test strip if the test strip gets wet. On the test site, we have a second antibody. We've called it antibody B, and that is fixed in position and cannot move from the test site. That antibody B is also attached with substrate at the other end. The third part is the control site, where we find antibody C, and again antibody C is fixed in position and antibody C has substrate there. Antibody C cannot move. Now in a pregnancy test, we will test for substances such as HCG, which is found in the urine of pregnant ladies and obviously not found in females who are not pregnant. And it is 2 specific antibody binding sites. One of those sites will bind with antibody A, and one of those sites will bind with antibody B. And that's the format for a pregnancy test. If we have a negative test, so if you do a pregnancy test and it comes up negative, this is what has happened. Firstly, the female needs to put a urine sample onto the testing strip. Once that happens, you will have antibody A, which again is free to move and has the enzyme at one end, but there will be no substance that we're testing for attached to that antibody. The antibody will then move along the test strip as the urine moves along the test strip. As we have no substance attached to antibody A at the reaction site, antibody A and antibody B do not interact. The urine continues to progress along to the control site, carrying antibody A with it, and antibody A will bind with antibody C at the control site. As a consequence, the enzyme that's attached to antibody A is now next to the substrate that has been attached to antibody C, and as a consequence we now have a substrate color change, whereas the enzyme's acting on the substrate. And as you can see, you have the control site has a dark strip in it, the test site has no strip in it, so this would be a negative pregnancy test. In a positive pregnancy test, this time we have the female urinate on the test strip. Once again, we have antibody A, only this time we have the CHG present and some of antibody A will attach to that substance. Just like before, the urine will move across the test strip and carry the antibody to the test site, but this time it carries the antibody A with no substance attached and some of the antibodies A that do have the substance attached. When they reach the test site, if antibody A has no substance attached, just like in the negative test, nothing happens. However, those antibodies A's that at the reaction site did attach to the substance in the urine will now attach to antibody B. When they attach to antibody B, the enzyme that is fixed on antibody A acts on the substrate that's attached to antibody B, and that means we will get a change in the color of the substrate. The urine sample then continues along to the control site. This time, all of antibody A that has any substance attached should have got stuck at the test site. There may be some that carries on to the control site, but we can simply ignore it because it doesn't come into play at the control site. The only thing we're worried about at the control site is the antibody A that has no substance attached, because that antibody A with no substance attached will attach to antibody C. That means the enzyme attached to antibody A is next to the substrate attached to antibody C, and we have a color change. In a positive test, we now have two test strip and the control site have changed color, and we have two colored lines on it. Now that's extremely important, that control site, because if we didn't have the control site, we wouldn't know if the reason for the change in the test site was because there's no substance attached to antibody A, or if something has gone wrong and antibody A has not actually got to the test site. By putting the control site after the test site, we know that the antibody A has made it all the way along the test strip, it has gone past the test site, and as a consequence, if the person was pregnant, then we would have had some sort of attachment at the test site. Now we do have some issues in terms of the use of monoclonal antibodies in ethics, and particular things like animal experimentation and the use of animals and monoclonal antibodies to come up with drug treatments. We also have an issue in terms of the monoclonal antibodies treatments have actually been linked to the deaths in some people. However, you could argue that the person who gets the treatment is probably going to die anyway, and as a consequence, this is a last-ditch help in order to try and save them. So there's an argument there. And also, again, we've got issues with drug trials in terms of healthy volunteers and the impact on those people. To finish off, think about the uses of monoclonal antibodies that we've discussed, and then think about the fact that it will cost animal life, and simply ask the question, do the benefits of the monoclonal antibodies and the uses we can put them to outweigh the cost of animal life?